Okay. So now we're talking about the cytoplasm. I encourage you guys to stay focused on our program. All right, what are we going back for? Okay, here we go. The cytosol, inside of the cytoplasm, inside of the cell are organelles. Non-membranous organelles are not enclosed by a membrane and always are in touch with the cytosol. Cytoskeleton, microvilli, centrioles, cilia, ribosomes, and proteasomes are like this. Membranous organelles are surrounded by their own lipid bilayer membrane, which is way cool. This includes the endoplasmic reticulum, Golgi apparatus, lysosomes, peroxisomes, and mitochondria. Here's another picture of our cell. What are the organelles? These are those little floaty things in there, separate compartments that have separate jobs. You can find this at figure 3.2. I'm not sure what page it's on. Let's just double check. You can see this picture on page 69. So basically what it's saying is there's two kinds. One kind of organelle with a membrane and one kind without. The nucleus, the Golgi apparatus, the mitochondria, they have a membrane. The lysosomes and the cilia, for example, don't have a membrane.
What about the endoplasmic reticulum?
I think it has a membrane. Let's just go back and check. Yeah, it has a membrane, endoplasmic reticulum. All right. The first organelle we're going to talk about is the cytoskeleton. You can see a picture of the cytoskeleton on page 75 on figure 3.5 if you like. The cytoskeleton acts as the cell's skeleton. It helps hold its shape and keep it functioning. It holds everything together. You can see it kind of right here in this picture as the cytoskeleton there. It's made out of microfilaments, intermediate filaments, and microtubules. And you can see pictures of that on, well, let's see if we got that picture here. Here it is.
Yeah.
Nice. Okay.
That's an electron microscope image.
That's right. And so it's all gray because there's no real color at that wavelength. It's so small. Well, in the human skeleton, there's over 200. There's not a particular bone in a cell.
No, but I'm saying they hold all that together, though.
No, not at all. This holds a cell together. It's like a skeleton itself. It's made up of microvilli. What is that? Those are just the wavy things that are on the outside of a cell. Here are the microfilaments, and the mitochondria are here. Yeah.
Right.
That's the concept I want you to know. One cytoskeleton holds it all together. Go ahead.
So how do they get this image? Do they chop the cell in half?
You know, I'm going to guess. What I think they did is they put the cell in a block of wax or a piece of wax, and they took a very, 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 very fine knife, and they cut it in half, and then they put it in the microscope.
Sweet.
Yep.
But what if like one of the the mitochondrion was off to the side and they didn't cut it in half, so you can see the insides of it?
Then you wouldn't see it if it wasn't cut in half. All right. So
That wonks.
it does.
Not even to see the mitochondrion.
Well, what they'll do is they'll make 100 slices and 100 images, and then you'll have kind of a 3D picture of the cell. So, all right. Notice that the filaments of the cytoskeleton have different thicknesses. Here we've got an intermediate filament because it's just a little bit thicker than the microfilaments, which are right here. Way cool. The shape of the cytoskeleton is determined by the genetic structure of the chromosomes. Different cells have different cytoskeletons, therefore different shapes. Go ahead,
Is the yellow
Andrew.
filament like a super filament?
You mean this one right here?
Yeah.
Let's find out. Microfilament, intermediate, microtubules, thick filament. I'd say that would be a thick filament right here, that yellow one.
Great.
Very cool. The next organelle to talk about are centrioles. They direct the movement of chromosomes during cell division. They organize the cytoskeleton, so the cytoskeleton is kind of built around them. And the cytoplasm surrounding the centrioles is called the centrosome. You can see a picture of the centrioles on page 77, I think.
In or on?
On page 77. So turn to page 77 right here. You can see a picture of the centrioles and what they look like.
I'm going to have to do some serious studying.
Kind of like this right here. Okay? There's two, and if you remember from biology, these two centrioles migrate to the opposite end of the cells,
Yes.
hook up with the cytoskeleton, line up the chromosomes, and pull them apart.
Metaphase?
I think so. Yeah. So,
So they can get in pretty close because that figure A thing right there is pretty darn close.
that's right. That is pretty darn close. They can get very close. I think this is a, well, I'm not sure. It might be a tunneling electron microscope or scanning electron microscope.
What's the difference?
One is over the top, one is inside.
I think that's right.
They stick the microscope inside the cells.
It's like a little needle.
Gotcha.
Okay.
So figure A is showing centrioles attached to the skeleton?
So in this case, figure 8 is only showing you a cross section of a centriole right here. Okay? Yeah. So that's a centriole. And its function, let's go back and take a look, is directs the movement of chromosomes during the cell division, organizes the cytoskeleton and the cytoplasm surrounding the centrioles and the centrosome. And it is anchored by a basal body, and oh, sorry, this is not centriole anymore. We'll find out in just a moment. A cilia is basically the little hair that wags around on the top of the cell.
Just like that?
Yep, kind of like that. It's anchored by a basal body and beats rhythmically to move fluids across the cell surface. Let's see if we can find a cilia.
All cells?
What's that?
All cells have?
Not all, most.
Google search.
For example, the cells lining the esophagus and the intestine and the lungs have that cilia there. Okay,
Can we see a Google image?
of a cilia?
Yeah, let's see it.
Okay, well, we're done for now. Let's just make sure, I want you to know that our quiz is going to be on reading up to 3-4, which includes the function of... Okay, that's too far. We're gonna stop on page 70. Well, what did I say?
77.
I said page.
That was uber cool.
I page 73. 3.3. Stopping at page 3, section 3.3. Okay, so not too bad. So it's not too bad.
I suppose we
So
feel
the organelles will not be in our quiz.
Let's do like one section at a time.
No, we've got a big book to get through.
that would be
We're not
more
going to get through the whole
efficient.
thing.
All right, so that's the end of our lecture. We're going to continue on with the rest of the organelles later. Thank you very much.